Thank you, Barbara. That sounded much more impressive in Spanish. I'm not sure what she said. But it's a pleasure to be here. American Airlines sends its regrets that I'm late. I had this terrible fear as the flight was delayed again this morning into Puerto, I'm not sure why, that the worst would happen. I would arrive here in time only to give my own talk. And that's like adding insult to injury. So I wanted to give you just a kind of a quick view of where we are with genetics and genomics, particularly in relationship to cetaceans. And I'm referring here to the molecular archive, the archive of DNA that each of us holds in every one of our cells, that contains not only our individual information, but information about the history of our relationships within our population and also, of course, of our species. And I borrowed this quote from Peter and Rosemary Grant. Some of you will know from their long-term study of the Galapagos, of Darwin's finches in the Galapagos Islands. And they are not geneticists. They are hardcore field biologists, ecologists. And yet they now realize it is a great time to be a geneticist. And I would add that it is a great time to be a cetacean geneticist, because I think all of us in the room would agree that whales and dolphins are not the easiest species to work on. And to have these molecular tools is giving us an access, a window, an opportunity that we never had before. And really all it takes is getting that little piece of skin now from a living whale with that biopsy dart that I'm holding. It's also because we've come through 2 centuries of intense exploitation of whales. This has reduced some, eliminated some populations, reduced others to very, very low numbers. In the case of right whales, we know from the century before last, 150,000 were killed, reducing the southern hemisphere population to perhaps only a few hundred. In the case of modern whaling, more than 2 million of the great whales, primarily bluefin and humpback. All of these reduced to very low numbers, creating a very interesting experiment in conservation biology and evolutionary genetics. If we look, here is just one example, the New Zealand southern right whale, the population that actually brought Europeans first to New Zealand for colonization shortly after Cook's original discovery for Europeans of New Zealand. We'll see that our current projection is that the population may have numbered between 30 and 45,000, much larger than probably was thought previously, but was reduced to very low numbers, perhaps less than 100, here by about 1912, due to first the intensive 19th century exploitation and then low levels of ongoing hunting. We think it's recovered to about 900 here in 1998 based on capture-recapture analyses of both photo identification and genotype identification. And we think that that's about doubled now over the last 10 years. So what can we learn from genes in time and space? Well, just about all the things we want to know, from individual identity, parentage, social organization, increasingly estimates of abundance from genotype capture-recapture, through to questions on an evolutionary time scale. Gene flow, that is successful migration of individuals from one population to another, long-term effective population sizes, genetic diversity, questions about subspecies and species. And then here in the middle, the kinds of questions many of us ask about conservation and management. What are stocks? How do we manage these? How do we conserve them? And I've used the example here of the butterflies, or annual species, but just to show you that a population is merely the connectivity of many individuals through their individual relationships over time. And the question we've all had to face over the last few years is whether we go wide or whether we go deep, to use a metaphor. Do we sample many individuals, or do we sample many individual genes from a few individuals? Because until recently, we really couldn't do both. So, if we could go wide and deep, many individuals, many genes, well, we'd be able to answer many questions Some that we cannot answer now, or some that we've not answered very precisely. Questions about population structure, but now at the kinship level, individual to individual. Effective population sizes, reconstructing bottlenecks, literally to the individual potentially, understanding the minimum size of the population or in men. To potential genetics or dynamics of recovery, the interaction of selection and drift. One is a neutral process, one is under some form of evolutionary selective pressure. Recombination, something we cannot do now. Genes that are closely linked to each other recombine at different rates, giving you a kind of an internal clock that can be useful for demographic reconstructions. Phylogenetics and taxonomy, we're still arguing over the sister relationship of blue and fin whales, blue and fin and humpback whales. Questions about subspecies. And hybridization, and I'll show you an example of this, the potential for genes to move from one species to another, some of which may be adaptive, some of which may be non-adaptive. I wanted to, just as a little nostalgia, remind you of what we used to do 20 years ago. We could take a biopsy sample and extract the DNA, and we would digest mitochondrial DNA and then visualize it through autoradiography, where we would have to label with a radioactive isotope a clonal probe and not amplify the mitochondrial DNA. This is not amplified mitochondrial DNA. This is extracted mitochondrial DNA that's been amplified visually through these radioactive isotopes. And we would look for little polymorphisms in the restriction fragment links. Here's a case where the mitochondrial DNA has been cut once, so it's a linear 16,000 base pair fragment, and then here it's been cut twice. So we know there's a polymorphism, there's a variant there. And when we put all those variants together, we could see from a relatively small sample of individuals from just 2 oceans that there's some pretty distinct differences in the haplotypes, the mitochondrial haplotypes of whales from the Gulf of Maine and the North Pacific. Well, no big surprise there. But within the North Pacific, also considerable differences. So this is an early case of narrow and shallow, but got kind of lucky, and it was an interesting story. It's still an interesting story, but we can go deeper now and we can go cheaper. Just a little history from restriction fragment linked polymorphisms. We went to sequencing, manual sequencing. Anyone who's done it never wants to do it again. Very tedious, but effective when it worked. To automated sequencing, the methodology that brought us the human genome. So the ABI 96-well capillary system was the workhorse of the Human Genome Project. That cost $1 billion roughly, took 10 years. The human genome, just as a reminder, is 3 billion base pairs in length. Next generation sequencing, I'm going to talk about 2 instruments briefly, the Roche 454 Junior, the Illumina. For shorthand, just think long read, short read. That's what it's about, is how long the sequence of DNA that these machines can read and how many times they can do it in a single run. So a Roche Junior, which is the small version, the lab top, bench top, can give you about 200,000 reads of about 500 base pairs in length. The Illumina, in one channel, one of 8 channels, can give you 20 million reads, but they're short. They were 40 to 60, now they're 80 to 100. But now, I'm projecting that by the end of this year there will be a whale genome. It may not be all stitched together and it may not be annotated, but the sequences themselves will be done because you're looking at basically one run on this machine, 8 lanes times this with some advances, to give you what we now think to be the 3.4 to 3.8 billion base pairs in the whale genome. And you could do that for $35,000. Not stitch it all together and annotate it. That'd take another year of an expensive postdoc or 2. But to just get the sequence, I think you could do it for $35,000. The efforts to sequence the whole genome of 10,000 vertebrates, including all the mammals, is underway. I'm aware of the bowhead being done by the Broad Institute, for reasons that will become apparent. The Beijing Genome Institute, one of the largest institutes in the world, is committed to do the baiji, the common minke whale, and the Chinese white dolphin. UC Santa Cruz has an initiative to do the humpback whale and the narwhal. But now what about going wide? I mean, it's wonderful to have a genome, but one genome does not answer our questions. So 3.4 billion base pairs of one individual will answer some interesting questions, but not some of the ones we'd like to know. So I just want to give you a sense of where we are with going wide, having adequate samples to address these questions, and I think we're doing pretty well. Southern right whales, 2,000 samples, primarily from the 4 calving grounds that are recovering in numbers. We're a little low over here, Nick, Simon. But I know that they're working to improve that. But we're missing many of the historical whaling grounds and essentially nothing from many of the feeding grounds. Humpbacks, the poster child of biopsy sampling, probably 6,000 was my quick addition. These mostly come from the South Pacific Whale Research Consortium for the Pacific here. Groups that Howard Rosenbaum has been involved with here, the Brazilians here, Marcia Engel and her group, very large sample numbers. Very large sample numbers from many of these breeding grounds, but still relatively modest samples from the feeding grounds. Here's the contribution of the SORP expedition here for Balleny Island. But otherwise, these IDCR samples are now pretty small number and pretty old. Blue whales, Antarctic blue whales, about 200 samples, as we'll see, not quite that many individuals, about 187 individuals. About 200 pygmy blue, at least that have been published. I wasn't able to get a full update on this, but I don't think the numbers changed too much, maybe a little larger here. Reasonable representation given the abundance of Antarctic blue whales, but nothing from their presumed breeding grounds, partly because, of course, we don't know where those breeding grounds are. But if we want to understand reproductive units, we have to sample on the breeding grounds. Oh, fin whales. Oof. Shocking. 750,000 fin whales were killed in the 20th century, according to whaling records. We seem to have only samples of 45. If there are more, I'd like to know about them. This is all I could get. This was courtesy of Eric and Archer and Barb Taylor. And I just wanted to make a pitch here. This is before I move on to the genomes. Is someone has to collect these samples, someone has to archive them, someone has to be generous enough to make them available. And we're not doing very much of that. There's a few groups who've worked very hard, and I want to commend the Southwest Fisheries Science Center for their collection and their willingness to share it, which is a lot of work. I would also say that we've worked hard to build a collection in New Zealand that is available as well on request for certain kinds of collaborative arrangements. We've got to do a better job of this. We've got to be thinking over the next decade or two about how to keep and make these available. So what about going wide and going deep? We have quite a lot of samples. We've got the genome technology to do that one genome. What about genomic approaches that give us a little bit of both? That means we've got to, in a sense, reduce our representation. We've got to reduce our cost. We've got to have population-sized numbers. So I'm going to talk about just 2 methods. Again, you can think of these as the short read and the long read method. We're using both of these to answer somewhat different questions and also to evaluate, how well they're going to work for us. One we refer to as rad tagging. It's a form of reduced representation shotgun sequencing, if that helps. Again, it's an Illumina-based short read. It relies on what's called de novo assembly of many short reads into gene fragments, and then usually used for analysis of single nucleotides polymorphism, so for discovering and describing SNPs. A second method is genomic amplicons and mitogenome resequencing, and we've been using the Roche 454, the long read, for this. This is PCR-based, which is a disadvantage, but it allows us to use whole genome amplified DNA, which is an advantage, and that is it allows us to preserve the original DNA through using this genomic amplified DNA. So the question we were asking with the rad tag was about the genetic similarity of the blue and the fin whale. And these are North Pacific blue and fin whales. In our example, we have only the rad tag for one each, but you'll see that that's an impressive amount of information. And our question was in general, but also we chose these two because these two populations do hybridize. That is, blue whales and fin whales in the North Pacific along the coast of California. There are several individuals that have been described as hybrids, and this is one of them. A fin whale-like dorsal surface swimming with a blue whale. And if John's here someplace, he could tell us more about this during discussion. This is a hybrid with a blue whale mother. It thinks it's a blue whale. It's never looked in a mirror. Its mother is a blue whale. It swims with blue whales, but it looks like a fin whale. So we were able to assemble 2.65 million base pairs of sequence from the fin, 1.5 million base pairs of sequence from the blue. These are assembled into gene fragments or contigs. We had about 7,000 from the fin. The DNA was a little better quality. These are on average about 365 base pairs long, so they're reasonable. The blue gave us about 4,000 of about 454. There are several questions that we can ask, but just in general, how many of those fragments are homologs? How many fragments from the fin whale are the same gene or gene fragment as in the blue? And in fact, we got a good overlap. About 3,000 of the fragments are likely homologs, that is the same gene. So we can compare them. So that's a 75% overlap. When we look at those 3,000 fragments, 1,200 of them are identical. There is no difference between these 300 or 400 base pair fragments of a blue whale and a fin whale. And then a few 800 differ by maybe 1 or 2 nucleotides, and then maybe 3, 4, but very, very few have any more than that. So maybe not too surprising, the blue and fin are very similar in terms of their genome. We would estimate 98.8% similar across more than a million base pairs of this comparison. And that's actually almost identical to the chimpanzee and the human. Genomic amplicon diversity, we chose to work on the world's smallest population of whales. Paul Wade's article on the North Pacific right whale, because of intense 19th century and whaling and 20th century illegal whaling, the current estimate of abundance is only 31 individuals. So I don't have time to give you much detail about what we've done with the nuclear genomes, but we have now sequenced the entire miter genome of every available North Pacific right whale sample. So we have 28 individuals, we have 9 different haplotypes, but you can see that there are some haplotypes that are shared by many individuals. And now when I say haplotype, I mean the whole miter genome. There is not a single base pair difference in the 16,000 base pairs of those 8 individuals. They are almost all certainly direct descendants of a female very recently. Diversity is much higher in the southern right whale. We're still working on the North Atlantic. So future directions looking to the past. I do have a minute. I want to just mention this because we have a poster about it. Here's another kind of archive. The bones of South Georgia Island and other whaling stations are a tremendous repository of this historical diversity. More than 170,000 humpback blue and fins were processed there, and their bones are available. Has that diversity been lost? Well, this is 223 samples that were successfully sequenced from 281 bones. Most were humpbacks, but a fair number of fin and a few blue. Almost every blue was different. So the haplotype frequencies as shown here in the pie diagram show you essentially, every individual has one mitochondrial a different mitochondrial haplotype. We can imagine that if you go from 200,000 or 300,000 down to less than a few hundred, that you will lose diversity. But still, a fair amount has survived. So in contemporary diversity of 187 individuals, there are 51 haplotypes. Only 5 of these are shared. We're still working on the question of whether that's a loss or not. Okay, conclusions. These are easy. Go wide. Get lots of samples. Go deep. Get as many genes and genomes as you can. The opportunities are there. It's taken another year or two before we can get, I think, to routine genomics of large population numbers, but it's happening. Reconstruct the past. We have great opportunity now for that. Assess the present. We have a great opportunity to understand that history of whaling and recovery. and all we can do is hope for the future. I'd like to thank our host, the government of Chile, our local convener, Barbara Galetti particularly, our absent coordinator, Debbie Steele, who's absent because of the birth of her new son just a couple of weeks ago, Kyle, the Australian Marine Mammal Center, whose enthusiasm and funding for this made it possible, my associates Beth Slickus and Angie Zarimba, and photographs from Paul Ensor and Brian Scarry. Thank you.